Don't break. Remember when
Hold it above your head.
Many of you don't know me, I'm Taylor, but I probably introduced myself. And we're going to share with you today an awesome presentation that we put together that is relating some experiments to biology concepts and then expanding it further into some more interesting things and how we can use it in daily life. So we can start yet? Okay. Well, welcome to the first annual Science Club Mixer, and we're probably going to try to have this tradition going for the next semesters and next years of new students. We are hosting it, the Bio Society, and then the Chem Club we're co-hosting. And first of all, what do you think that humans, fruits and plants, and viruses all have in common? Anyone take a guess? Yes, DNA. You're awesome. You got it? Okay. So, this is an amazing molecule, and we're going to explain what it is. So, what is DNA exactly? What is this thing? Well, it's polymer of a bunch of little monomers called nucleotides, and it's very tiny. So, we're going to show you how small it is in relativity to other things in the universe and common everyday objects we see. This is called the scale of the universe, and when it loads, we're going to show you. And it's this pretty cool thing that zooms in and zooms out, and it has different items that are size scaled to each other. Unfortunately, this computer is really slow. Because in Longitude Hall, there's computers that are very slow in every class. But anyway, how are y'all doing today? Good. Good? How you doing? I'm doing good. It's really cold out suddenly, isn't it? Yeah. Okay, so there's a human. There's a flower, a peach ball. Through this little mouse, like animal. There's an ant. There's a dust mite, paramecium, human egg cell, and a cell nucleus. There's a chromosome. Does anyone know what a chromosome is made of? DNA. And it's just a condensed form of DNA. And way down is a molecule of DNA. And then go further, you can see some atoms, and there's a phospholipid. And there's a water molecule, and a protein. And now you see how really, really tiny DNA is compared to everything else. Even though these pictures blow it up and make it look so huge, but it's so tiny. Well, the structure of DNA, it's a double helical structure, and it has two strands. And their backbones are made up of phosphodiester bonds. They're connected, and then there's ribose sugars, which are 5 carbon sugars, with a phosphate group attached to the 5' end, and a hydroxyl group attached to the 3' end. And the two strands of DNA run in opposite directions, called anti-parallel conformation. And that means one strand goes in the 5' to 3' end direction, and then the other strand goes in the 3 to 5 direction. And on each ribose sugar, there is a nitrogen base attached, and there's adenine, guanine, cytosine, and thymine, and they're all the bases. And they complementarily pair together. The adenine and thymine bond together with 2 hydrogen bonds. The guanine and cytosine bond together with 3 hydrogen bonds. And they alternate, every person has, and every organism has their own unique sequence of bases. And just keep in mind, do you guys know the charge of the overall charge of DNA? Positive. Negative, yes. And just keep that in mind for the rest of the presentation. Okay. So now the experiment we're doing today is the DNA extraction from a banana. Does anyone know how we can get DNA out of a banana? What we have to do to get it out? Well, what did you? Smash it first? Yeah, that's the first step of the experiment. Now, someone is opening a container that has banana in it to keep it fresh. And we're going to start the experiment by taking half of the banana. And we need some volunteers, at least 2 volunteers to help Sonny out so we can have some hands-on experience on this. Anyone want to volunteer? It's really easy. You can come up. Anyone else? Okay. Wendy, come over. That's it. you want my lab goggles? I have a pair. Number one last thing, the banana. And put the banana in the bag first and mash it up into a very liquidy slurry of banana. Now, what do you think that the purpose of mushing up the banana is in this experiment? Since we're trying to get the DNA out of the banana. Does anyone have an idea? When we mush up the banana, it loosens up the tissue and the cells, and it pretty much makes it easier to access what is inside of the cell, the DNA. And as we do this, it keeps loosening and loosening. And then right to the next one of these And the supplies of this experiment are clear cups, stirrers, plastic bags, some coffee filters, and just simple things that you can find around your house. So do try this experiment at home. When you're not busy, you could show it off to your grandparents at Thanksgiving. You could say, hey guys, I can take some DNA out of the banana. The materials in this experiment are isopropyl alcohol, which you probably all know as rubbing alcohol, and distilled water, salt, soap, and bananas, of course. And there's a little cartoon for the DNA coming out of the banana. Well, where do you think the DNA is located in the banana? Does anyone know where the DNA is? Yes. And the nucleus is within a cell. And bananas are made up of many cells. And the cell is made on the outside, it is surrounded by a membrane. And so is the nucleus, it's surrounded by a membrane. And they're both membranes, and all biological membranes in general, are made up of this certain type of material. A phospholipid bilayer. It's two layers of these little phospholipids. The head of the phospholipid is hydrophilic, which means it likes water. And the tails of the phospholipid are hydrophobic, which means they don't like to be near water or an aqueous environment. And the two layers face each other in the phospholipid bilayer, so the tails are on the inside avoiding water, and the heads are on the outside with the water. And what else? Well, the head is polar because polar pretty much interacts with the polar water, and then the tails are not polar, and they like to avoid the water. And when we are doing this experiment, the phospholipid bilayer is very important in the method of taking the DNA out of the nucleus. And here's a little model, just like the real phospholipid bilayer. There's the heads, the hydrophilic heads, and the hydrophobic tails. And here's the aqueous environment that surrounds it. So what can we do right now?
We are adding GI water, which is making the...
Well, now we're making a banana and water solution. We're putting deionized water in the banana in the bag, just to make it more liquidy. And we're not at this point yet. We're going to add some soap to the bananas. Can anyone tell me why you think that soap is a good thing to add to the banana mixture? And why it would play a part in this experiment? From experience, have any of you did this experiment before? Or heard of it? No? Are you doing soap?
Yes.
Okay.
We're going to add the banana slurry to the soap.
Okay. So now we have some soap, dishwasher detergent, in one cup, and then the banana mixture in another cup. We're going to add them together and mix it. And we're adding detergent to the cells to open up the membrane and to also open up the nuclear membrane, so that the DNA can slip out into the outside environment. And how does the detergent actually open the cell membrane? Well, the phospholipids in the soap interact with the phospholipids in the bilayer, and it causes a disturbance in the membrane. And the bilayer opens up. The phospholipids link together in single membrane conformations called micelles, which has no water on the inside, so the tails are happy because they're hydrophobic and they don't have to deal with the water on the outside. It creates a little space in the membrane so the DNA can slip out of the nuclear membrane and then slip out of the cell membrane into the exterior environment. And there's the DNA. This is, well, this is after the experiment's done, that's a complex of DNA and proteins that floats to the top of the water and alcohol and soap mixture. So it clumps together and forms a little web-like structure. And that is the DNA and the proteins that are associated with it from the banana. So after the students finish with the experiment, you're going to be able to come up there and look at it and actually see what it looks like in real life. So where are you guys thinking this?
We are mixing the banana and we're breaking the cell membrane.
Right now, they're breaking the cell membrane of the banana, sorry. And they're exposing the DNA from the cells. And should I continue? Well, why do you think we're actually trying to get the DNA out of a cell? What do you think we can do with this molecule? Why is it so important? Why do we even care? Well, have you guys ever seen crime-themed shows like NCIS, CSI, Homeland? Yeah? And what do you think they do to find the criminal? What kind of thing do they do?
They look for fingerprints or any material left from the scenes.
Yeah, exactly. And they take the samples from the scene. They take it to see the DNA of the person that left it. And they're going to use it in a special process that we use in laboratories called gel electrophoresis. And what exactly is this? Well, there's a box called a chamber, and each end has a charge, a part of a cord plugged in, connected to a power source. One end is called an anode, which has a positive charge, and the other end has a negative charge called a cathode. And within this chamber is a gel, which is like a rectangular molded agarose medium that it's like a little jello conformation. It's liquid when it's heated, and then it solidifies when you put it into room temperature. And there's wells at the head of the gel in the negative part, and that's where you insert the DNA samples. And how does this work? Well, we use special enzymes called restriction enzymes. And since the DNA molecules are so big, do you really think that a long molecule can fit into one gel it wouldn't really migrate that efficiently. So we use the restriction enzymes to cut out the restriction sites, which are specific to each person or organism, and create large and small, different size length fragments in base pairs. And then we inject these, we add these into the wells of the gel. And the wells are at the negative side of the gel. So why do you think that the wells are on the negative side? What does this have to do with DNA's characteristics? Does anyone remember what I told you in the beginning of the presentation? Yes, DNA has a negative charge. So negative charges repel each other, and the positive charge would attract the negative. And as the DNA migrates through the agarose gel, it will be slipping through the pores, since it's a porous medium. And the smaller ones, what do you think happens to the smaller fragments? Do they move further or not as far as the big ones?
Not as far.
Yeah, because since they're small, they're lighter, and they're easier to move and maneuver through the porous gel. And the larger molecule fragments are left behind. And it creates a unique banding pattern depending on the restriction sites in this DNA molecule, per person or per organism. In this situation, we're using with humans for crime scene analysis. And then now, I'm going to put you guys to the test and see if you can find who committed the crime. You have to compare the known lane of the gel, the evidence from the crime scene, to the various suspects or people that are involved in the crime. So let's see who did this crime. Take a moment to figure it out. Yes, you're right. It was suspect 2. And then here's another one. We have some Disney characters and Plankton. I don't know why Plankton slipped himself in there. He's a little clever thing. And we'll have to see who committed this crime. Take a moment to figure this out. I want to see who can figure it out. Remember, you have to match the known. Let's see if you're right. Yes, it was Ursula. She was the criminal in this situation. And now another way you could use DNA testing. Have any of you guys ever seen Jared Skinner? Yes, you have. So, well, how do you think they figure out who's the dad of this child? They can't just pull it out of thin air, can they?
They do a DNA test.
Yes, they do a DNA test. And what are these DNA tests actually? What's the science behind it? You can't just pull DNA from a guy and say, oh yeah, this is the guy. Well, you see how blue and yellow can't make orange. They make green. But that's his, they have to do a DNA test to figure out who's his dad is. Now figure out which cat or skunk is the father of the twins. Compare the dads with the child. What did you say? Felix? Let's see if you're right. Yeah, it was Felix. He was the dad of the little kitten. And here's some comics you guys can feel free to look at. That's what something else. That's what a retrovirus looks like. Did you not know that? And then take a moment to read this and see if you can figure out what it is.
Who is that?
Yeah, who is this guy with curly hair? Is it Watson or Crick? It's Watson, that's James Watson. And what do you guys know about James Watson? Does anyone know who he is? Do you know who he is?
We discovered DNA?
Yeah, they came up with the structure of DNA. Yes, the double helical structure, right. And what kind of what laboratory do you know if they work in? It's pretty close to home. You would never know. It was in Cold Spring Harbor Laboratories, and that's in Long Island, New York. So if you guys are interested, the Bio Society is going on a trip to Cold Spring Harbor Laboratories where James Watson actually resides, and we saw him last year when we went on the trip, driving in his fancy little Jaguar. And we're going to go on this trip in the spring. Let me know if you're interested. We'll have a sign-up sheet for that trip, and then we also have a trip to the Museum of Natural History in New York City coming up on February 21st. So if you want to sign up, you don't have to be a bio major. You can join the Bio Society on Campus Cruiser, and you can put your name down on the list for the trip. And it's $15 for bus included, food included, and admission. So isn't that a great deal? That's a great deal. Don't tell me no. And if you guys want to see the experiment so you can show your friends and show your family at Thanksgiving dinner, here's a link if you want to take a picture of it or come up to the slideshow at the end. And how does DNA fingerprinting work? That's another thing you can look at on your own. It shows the process of DNA fingerprinting. And here's some source to use in the experiment. There's a collage of pictures we took. That's the beach suite from last year. We went to Lawrence Harbor and picked up some litter and cleaned it together. There's some members of the Bio Society board from last year. There's Fabio, I think he was the treasurer. There's us, this cast, Student Activities Fair. There's another photo we took. And another table we had at another Student Activities Fair. So if you want to be part of the fun, join the Bio Society and you won't be disappointed. Trust me. And Professor Gardner was kind enough to come and support us. He's our advisor.
Great job.
Here's some social media that you can connect with us on. Take a picture of it, plug it into your phone. Like us on Facebook, because you'll get the updates quicker than Campus Cruiser.
Great job.
Thank you. Stay tuned, we're going to have some other guests. Glenn Fernando and Alex is going to do some experiments.
Nice. No, not that one. What are you doing? All right.
And you can pull it out.
Yeah, exactly. Yeah. Nice. That's what's in your cells. Ooh. Look at that. You want some? You hungry? It's banana flavor. Good job. Did you guys join the Bio Society?
My experiment is basically composed of two different molecules, which I want to show the differences in polarity. Basically, water, as we all know, is composed of oxygen and hydrogen, but it's actually polar. And the polarity is due to the bonds and to the angle. And that's what makes water very polar. And it is the intermolecular between molecules are hydrogen bonds. While water has that fluidity. And my experiment is basically doing a lava lamp in both the water and oil. Basically, I'm gonna put the oil here first. And this is basically vegetable oil. So the molecules of vegetable oil, well, you can't really see it from this thing, but here's a glycerol composed of 3 fatty acids. Now, what makes this molecule non-polar is the change of carbon. So hydrogen can be composed from anywhere between 14 to 20 carbon bonds, and that's what makes the oil non-polar. What I'm going to do next is I'm going to add some water. And as you can see, the water and the oil are beginning to separate. And that's basically because like goes to like, and because of the water being polar and the oil being non-polar, they start to make the two solutions start separating each other. I just want to get a little bit higher. And I'm going to add some food coloring to give it color. And this is basically polar, so you can see it how it's going down to the water. It doesn't bond with the oil. It doesn't remain there. This is Alka-Seltzer. Now, when I add the Alka-Seltzer, what it's going to create a carbon dioxide gas, and the carbon dioxide is going to bubble up, creating a type of lava gas, I would say. And here you can see the bubbles going up and down. That's something pretty cool that you can see. So I mean, yeah, here they have the phone. That's why I use my phone. Yeah. What I'm going to do is I'm going to show you how it looks.
That's really awesome.
Yeah, so that's what it's supposed to look. So that's basically what it creates. It's how the molecules work when they come in contact. They're not polar. They're not. The difference in polarities will not allow the two molecules to bond, creating this mixture. And the bubbles are being formed from the carbon dioxide being released from the alcohol. So, yeah. Thank you, guys.
Chemistry club on site, and today I'm going to be doing a pigment extraction. And